Next slide. The top of that slide should say concept KBase user experience. One of the things that we're working on is a new style of user interface. Of course, KBase has a programming interface. It has a website based interface, but we're working on this new kind of interface that is sort of a cross between an interactive notebook kind of interface, might appear to be somewhat similar to things like Mathematica or MATLAB or IPython, that has bound to the environment the API, so you can do commands and so forth. You can share visualization, but also has coupled to it this notion of collaborating and sharing information. Next slide, please. And one of the reasons for this is allowing teams to actually flexibly collaborate in doing data analysis and doing modeling, and to share information in small groups, and to track which methods are actually being used. Next slide. This is the schedule. We've had 2 releases so far. We're working on a beta release in August that will have early adopters to experiment with it. If you're interested in trying this out, get in touch with me or go to the KBase website and send email to support. This whole environment, the integrated environment, is scheduled for release in February 2013. Next slide. I think I'm going to, I'll just give you 1 or 2 of these slides. One of the things that we've learned over the years in building these kinds of systems is that the way you manage systems for data-intensive computing is quite different from how you manage systems for simulation. In this chart, data-intensive computing, usage models chart, sort of just indicates what we've learned. I mean, data-intensive systems need continuous access based on the rate of data submission. So if we have people submitting a lot of genomes to our annotation server, we need to keep scaling up the back end for some period of time, and then we might have to, if we don't have then sustained demand, we can scale that back, but the system has to be very responsive to the user load. CPUs, I/O, data storage, all are important, and really allocating them doesn't really make sense in any way other than to keep up with demand. The data products that are produced are typically passed from one stage to the next in some kind of a pipeline, and that means that you have to take care in designing the intermediate data products such that they can be ingested. And the other thing that's really quite different is the data products from analysis are often made available for other external users to analyze and use, and they're curated over time. And you can compare that to batch-oriented simulation, and what you discover is that you really need, you might have very similar infrastructure, but you have a very different management model. So I'm just going to skip forward. I think I'll skip to this slide called What is Important, and then I'm going to end after this, Jeffrey. So people are often fixated on data volume, and I sort of want to curb everybody's enthusiasm about that. What's really important here is not the data volume. It's whether or not we're able to extract information from the data, and whether or not what we're learning, that is information and knowledge, is increasing as a function of the data volume. And that's not so clear. I mean, we're doing a lot of sequencing, but a lot of sequencing is resequencing, right? And so what you learn upon, say, sequencing the 30,000th human genome was not nearly as much as when you sequenced the first genome, right? So you might have a lot of data, but you're really not generating a lot of information. It's true that biology will have large amounts of data, but so will many, many other fields, and biology really isn't really driving the data volume. Where I think it's really driving things is in complexity, or how one handles errors, both in the underlying data sets, how you resolve inconsistencies, and how you reconcile data and incomplete knowledge. That's how you really tie the knowledge back into the data. And these are at least as challenging from an algorithm standpoint, from a science standpoint, as dealing with sort of the scale. And I mentioned this notion that the raw data is increasing faster than we're closing the loop on understanding. That means 2 things. One is that the algorithms we use for analyzing data today are going to be quite different from what we're using tomorrow, because every time we learn something new, we want to change the analysis. That's sort of clear. But also that we might want to change how we're collecting data based on understanding. And so there's a challenge to make sure that we keep in the forefront, sort of the scientific goal of what we're trying to accomplish. The other thing that this means is that as data volumes increase and become cheaper, often it's more cost effective to rerun an entire experiment on a current technology, say sequencer, than it is to try to reconcile older data. So I think with that, let me just skip to the acknowledgments. What I talked about here is supported by DOE, NSF, NIH. We've had grants in the past from DoD. Of course, Argonne and Chicago put this in. We've had a lot of foundation support, and everybody has been very, very appreciative of the support. And next slide for acknowledgments is that, what I reported on here is the work of many, many people, and I've highlighted some of them here. I encourage you, if you're interested in this kind of a project, to get in touch with us. We have lots of opportunities for people to collaborate and engage. So with that, thank you.
Thank you very much indeed. Is my mic things. So let's first have the virtual applause. And now we can have the virtual questions. And we have one over there. You can rush up here. We have a question from Indiana.
So when you were talking about, I guess, attempting these metabolic reconstructions from metagenomic data sets,
yep.
Do you think the current sequencing technology is kind of useless in that regard, in that we're attempting to reconstruct from short reads, so it's difficult, I guess, to assemble from individual genomes from that? And do you think things like Oxford Nanopore are actually going to really help in this regard, like long read sets?
Okay, so I would say that the challenge of short reads for metagenome assembly has caused a lot of people to work on assembly algorithms, including us, and I think that we're making good progress on being able to do pretty good assemblies from short reads. However, things like Oxford Nanopore are going to change the game. If you can get single reads that are 10K or 100K long, where we understand the error model, and of course we don't yet know what Oxford's error model is going to be, but we know in iTorrent, it's different kinds of errors, indels instead of SNPs and so forth. What is interesting about metagenomics is you don't always want long reads. That is, metagenomics is very similar to sampling in population biology, and often the number of samples is equally important to the quality of individual samples. So the problem with something like very, very long reads is that you're most likely to get hits in the abundant part of the community, of the rank abundance curve, right? And so if you just did really long reads, you get a lot of reads of a relatively few diverse organisms. Whereas if you had a very, very large number of reads, even if they're much smaller, you're much more likely to sample the tail of distribution. So it depends on the question how useful these long read technologies will be. But having said that, they're going to be great. It's going to be a whole lot of fun.
Thank you.
So, Rick, I had a question. You showed these 3 data stores.
Yes.
I forget the exact names now, and one of them you said was using MongoDB.
Right, that's what we call the persistent store. That's for the user workspace.
So what were the other stores using? What technologies?
Okay, so the central data model, which is our tightly coupled, integrated public data repository, that's using MySQL. And the bulk data store is essentially using it's using Mongo, but Mongo is only for the metadata. And then we have a parallel file system that actually supports the bulk data. And so there you have a web interface, Mongo for metadata management, and then a massively large parallel file system that supports high performance parallel transfers. And so you use the Mongo in that context for setting up control channels, and then you use something like Globus Online to make very large scale high bandwidth transfers to and from sources.
So you're not using object stores, you're using actually things like Lustre or GPFS?
Well, we have a system called Shock that integrates a layer on top of it, but we're using, we're not using an object database. We're using what amounts to the same strategy that we've used for a long time, for large-scale data, which is a combination of a database for handling metadata and sort of containers, and then a high-performance file system that's pointed to by that database for the actual objects. Because in the context of like these metagenomics data sets, you might have a terabyte as a single thing, and you have metadata about that, but you don't have any need for much internal structure of that object. So we treat them as sort of just undifferentiated, binary stuff, blobs, right?
Okay, thank you.